Es un honor participar en este congreso y agradezco a los organizadores soy muy contento de presentar nuestra investigación hoy. Gracias. I want to tell you the story of a 25-year collaboration with Francisco Lopera, my very close friend and collaborator in Colombia, where we have found the largest family in the world with a form of Alzheimer's, a genetic form of Alzheimer's, a heritable form of Alzheimer's. And these people live in a remote area in the state of Antioquia, which you see on the map here. The capital of Antioquia is Medellin, right in the center, the yellow dot. And the family lives both in Medellin, and many of them come originally from the campo, from the countryside, out in these in the mountains around the city. And just give me a moment here. Let's try this one. I am probably a little technologically challenged. There we go. Okay. So this family, as I say, is the largest in the world with familial Alzheimer's. Here you see just a few of the pedigrees. This family, if we put the entire kindred of 5,000 individuals on the walls here, it would more than fill this room. And these are very, very large families with a form of dementia that begins like clockwork at age 45 and progresses over the next 10 years to death. It's inherited as an autosomal dominant trait. That means 50% of the offspring will get the disease. And because the families are so large, the 10, 12, 15 children, very commonly, this disease has actually spread quite rapidly through the state of Antioquia. As I say, these are just some of the families. And one of the, when we first began to study this kindred, we didn't know what the disease was. We knew it was inherited. We thought it was Alzheimer's. We hadn't yet proven it. And we suspected a gene was involved, but we hadn't yet discovered it. And so we, though made progress over the ensuing decade by collecting lots of samples, eventually in collaboration with a group in St. Louis and Allison Goat, we found that there was a mutation in this, in presenilin, and it is a particular mutation here called E280A. Like all the mutations in presenilin, this is what's called a private mutation. It's only present in this one very large family, and it is like the other mutations, they're all confined to just single families. And this one, as I say, is unique for the family in Colombia. Now, we wanted to trace the origins of this mutation. This mutation, by the way, is well known to cause Alzheimer's. That is, mutations in this gene are well known to cause Alzheimer's. There are quite a few mutations in other families in the same gene, all leading to the same Alzheimer phenotype. And we were interested in the origin of this gene and traced the origins of the immigrants who came from here, from Spain, through Darien Bay up here, and migrated down through this very rugged terrain. The Sierras here are really have steep ravines. It's intensely overgrown countryside even today, and found their way here to what was the original capital of Antioquia called Santa Fe de Antioquia. Today it's Medellin. Back then this was the capital, and there were several routes that the Spanish conquistadors had followed. Now, we know that coming through that route was really remarkable because this area where a number of the conquistadors arrived after 1492 in many, many boatloads of individuals, of males who came over from here and came through territory that is hard to imagine that in less than 50 years they reached Santa Fe de Antioquia, which is in the interior, and made that the capital. Because, as I say, to travel in this region today is really, really difficult. The Pan American Highway ends in Panama, and it doesn't pick up again until you're far into South America. And this territory is nearly impassable. But nevertheless, they established a number of towns and laid down the foundation for an admixture genetics, a mixture of Spanish males and the Amerindian females in numerous waves of migration, leaving today the genetic imprint of this admixture. And we see the history in the genetics because when you look at the Colombians from this region, on their X chromosome, we see the Amerindian genetic markers. I'm sorry, on the mitochondria, we see the Amerindian genetic markers of females, and on the Y chromosome, we see the markers from the Iberian Peninsula. And I'm going to take you briefly on a short tour of the kind of work we do to introduce our studies. This goes back quite a few years, where we would gather families together, in this case in a hospital in Medellin, where Dr. Lopera and myself have called some of the families in to do what we always do, which is to talk to them, explain the disease, dispel any myths that they have, and collect samples and enlist their participation in these studies. I like to show this picture because in Colombia there are no nursing homes, no assisted living facilities for elders. So as the people with Alzheimer's get older, they live with their families until they pass away, until they die. And there is not the kind of barrier that you see in the United States between older people and younger people. They are right through till death. The young are very, very close to the people with Alzheimer's. There's no warehousing, as we say, of these individuals. The people with Alzheimer's from these families live in these barrios around Medellin, in these hillsides, from a rather poor area. They've migrated in from the countryside. And here's an example of us visiting one family. This woman has advanced Alzheimer's disease. In contrast to most of the families with very large numbers of children, this family is very small. In fact, this girl who's 17 years old has been taking care of her vegetative mother for the past 9 years, almost on her own in this shanty region. And this is our nurse, Lucia Madrigal, who is a remarkable woman. Those 5,000 individuals in that family, she knows every single one and has the entire pedigree in her head. From here, we traveled out to Yarumal, which is about a 5 or 6 hour ride. We leave very early in the morning and go winding through the mountains. And here's the plaza in Yarumal. And again, call the people together to explain what's happening in their families and to have them understand a little bit about genetics. This gentleman, when I was out there one time, this gentleman stood up and asked, he said to me, well, you're telling us we have this genetic disorder. Is it okay if we marry our cousins? And as a geneticist, I said, I don't think it's a very good idea. And then he said to me something that I could not answer. He said, but doctor, we have no one else to marry. And I did not have an answer for that. Some of the people live in quite remote areas who are participating in this study. This is a family who gathered with us in Sopetran, and they don't live in Sopetran. They actually live in another town called Moron, which has no roads. It's on the other side of this mountain, and they came in by horseback to gather here and meet us. There's Lucia in the middle. And we have a social program as well in Colombia, again, run by Dr. Lopera, where if someone has certain needs, we actually ask them what their needs are. And the number one need that they asked for was diapers. But we also have provided hospital beds. And on one occasion, we drove quite a distance all the way out to this town along this road here to a town called Betania, where to pick up a bed from a woman that had recently died of Alzheimer's and to bring it back into one of the barrios outside of Medellin called Manrique. And in this, the reason I'm showing this slide is because there's another contrast in Colombia to at least the way we deal with Alzheimer's and medical issues in the States. And that is this issue of privacy. We certainly we honor privacy, we respect privacy, we maintain the importance of privacy about ourselves. But in these settings, there's very little privacy. The walls between the house and the street are very, very thin. Everybody knows what everybody else is doing. And by the time we got back to Manrique with this bed, it was after 10 o'clock at night, and the entire neighborhood gathered to join us in the delivery of the bed. These people here, none of them are related to this family. They're all in the apartment watching the delivery of the bed, and everybody is interested in what's going on in the neighborhood. Privacy is really something that doesn't exist in the same way we're used to it. And finally, I'm going to take you here to another town called Angostura. This is a place where my colleagues did not let me go until recently. There's still some trouble there. You see the military guy. But it was a very important place to get to because there are a lot of families that live there. And once again, we gathered the families together and discussed with them the various issues and concerns. They have all kinds of notions and myths about the origin of this disease. Some have asked, they said, well, we believe it comes from touching a tree, but we don't know where it comes, which tree it is. Others believe it comes from touching someone who has the disease, and a part of our job is to destigmatize those kinds of myths. So all of that has resulted, I think, in us trying to bring a knowledge to the population and a maturation of their thinking so that we can prepare them for what is about to happen actually now, which is the first prevention trial for Alzheimer's disease. As you know, in Alzheimer's disease, there has been a number of phase 3 failures of drugs. And the thinking in the field has actually progressed to the idea that now we have to treat this disease before it strikes to make a difference. Because if we start treating once the person has the disease, it's probably too late. So at this point, after working in obscurity for well over 20 years with this family, this is when the Colombian family began to get international attention. And why? Because in this family, we know who's going to get it. They have a mutation. We can easily test. We can even test an unborn child in a mother's womb for Alzheimer's. We don't do that, but it's possible. And they get dementia like clockwork at age 49. They get mild cognitive impairment at age 44 or 45. And the disease progresses, like I say, in a very clock-like manner. Now, here's another reason why it's become possible to do a prevention trial here. In this study with PET amyloid, a fluor beta pair done by Adam Fleischer in Banner, we brought to Arizona a number of members of this family to get this amyloid scan to detect amyloid in the brain of living individuals. And here's the typical pattern. The yellow and the red is the amyloid. There's lots of it in Alzheimer's disease. There's lots of it. Even in MCI, there's lots of amyloid. But importantly, here, these people were getting amyloid in their brain at age 30, even a few in their late 20s. So when we talk about a prevention trial here, we're talking about giving a drug, in this case it's going to be a monoclonal antibody against amyloid called crenezumab, to these individuals here before they get symptoms. However, this is what we would call a secondary prevention trial because they already have some amyloid in their brain. And therefore, to treat as a true primary prevention trial, we would have to begin treating when they were teenagers before they had any amyloid at all. I will, before I mention this slide, during the questions I can answer more about this prevention trial, about how it's designed, and more details about the drug. There's been a lot that you can read about the trial. The first people to get drug will happen before the end of this year, and we're very excited about that. Now, what I'd like to talk about today is really the genetics behind our work. For many years, this large family with a lot of consanguinity, we were quite surprised to never have detected any individual who was homozygous. And we thought that homozygosity of presenilin mutations would be lethal. However, recently, we discovered the first homozygote. Here's the mother, here's a mother and a father who both carry the mutations from two different branches of the family. And one of their children here is homozygous for presenilin. This is an 8-year-old girl. I've met her. She's charming. She speaks nicely. She makes eye contact. Clearly, homozygosity of presenilin is not lethal. We don't know what's going to happen to her in the ensuing years. I should say, though, that we have been just, if anybody's wondering about the ethics of the mutation testing, because she was of exceptional medical interest, we presented this case to several ethical committees to ask if we could anonymously test her for this mutation, and with the proper protections in place for confidentiality, there was an agreement that she could be tested. So to move into more detail now, to move to the more scientific part of my presentation, I want to talk to you about the whole genome sequencing that we're doing on 116 individuals within these pedigrees. And the first thing that we saw was that here is the chromosome 14 where presenilin resides and where the mutation is. And when we looked at the sequences from all of these individuals with the mutation, all of their DNA in this region could be aligned, some with longer pieces and some would be with shorter pieces. But in addition to all having the mutation, they also all had a stretch of DNA which was identical among all 116 of these individuals. Some of them, I say, very long, some shorter because there's recombination going on, but there was this length of DNA called this identical by descent region, or IBD, and that is unequivocal evidence for a founder effect. There's no way that this could have arisen in any other way but by a single individual at some point in history leaving traces in his offspring with this mutation. Now, I say his offspring because we also know that the markers on here come from markers from the Iberian Peninsula, from here. So that the local ancestry estimates confirm that the mutation is of Mediterranean origin. Furthermore, we can tell by the amount of recombination, the shifting that's going on from generation to generation through 18 generations, that this mutation must have occurred sometime around 1550, around the time when the Spanish, shortly after the time the Spanish arrived in that region of the world. So the question that we now posed was whether or not we can find any genes that would modify this age of onset. Right here, could shift that. And we know that almost everybody, as I mentioned, has an age of onset right around age 49. But there are a few outliers. People out here fall far outside the mean. This must be a strong allele to push the age of onset this far out. And it would be a protective allele, which we'd very much like to know what it is. And these people have a deleterious allele, which is making their disease come on earlier than the mean. So these are the people we're really looking for some variant. Well, we've sequenced over 100 genomes, and we don't have the results yet, but we have preliminary results from 6 individuals, 2 with a deleterious modifier, 2 with onset around the mean, and 2 with a protective modifier. And this, the data I'll show you here is, and I will just make this point very, very overtly, this is non-statistical data. This is just a little taste to see the kinds of things that we can do because it's a very small sample. But if you make binary comparisons between each of those 6 individuals and lay out their DNA and show where they're common between each of the 6, and on the outside here are the chromosomes, and each one of these concentric circles is 2 individuals compared to each other. You saw there was a father and a son. That's this circle where where a lot of their DNA is very similar. Here's chromosome 14, where all of their DNA matches up. And what we're looking for are places like this, where 2 individuals who both have the protective allele have a same stretch of DNA. Somewhere in there could lie a protective gene. And indeed, in one of those places, we found a variant of a gene that's in the presenilin complex. It's called AF1 and is part of the gamma secretase complex that cuts A beta and makes the amyloid peptide. There is a variant, a genetic variant in there among 2 individuals who actually have a delayed onset. Now, as I said, that's not statistical, but it gives us a first look at a gene which could be biologically implicated in this delay. That is somehow modifying the effects of presenilin mutations to make it less toxic. So let me mention, though, a few issues that we face here in dealing with this kind of work. Because even though there are a lot of people there in this Colombian kindred, big enough to do a drug trial, it may not be big enough to get a really true genetic statistical sample. Sample size is really an issue if we want to try to find any statistical association with this disease. It's been a real challenge. And there are other issues too. That is, if there's a modifier gene, how frequently is that modifier gene present in the population? Is it a common variant or very rare? And what is the effect size of the modifier? Is the modifier going to really make this disease much better, or is it just going to tweak it a tiny little bit? And do we really have accurate phenotypic information? So with Alzheimer's, we call a lot of people Alzheimer's disease. How do we know they really have Alzheimer's disease? We're better at it now because we have the amyloid PET. But if we make a mistake in our diagnosis, then some of this phenotypic information is going to obscure our genetic conclusions. So one of the ways in which we believe we can actually enhance genomic data with small samples is to do superimpose upon that technique a second technique. Now, I'm sure you're familiar here with this technique called human-induced pluripotent stem cells, in which you can take a biopsy from the skin of anyone in this room put those cells into a dish and turn your cells into your very own stem cells that have your genome growing in the dish. We can then make those cells into neurons, and in so doing, we now have neurons from a specific individual growing in the dish. We could never do that before. How could we ever get at the genes expressed in the brain without doing a brain biopsy? Now all we have to do is do a skin biopsy. And to do that, we basically plate these cells, turn them into stem cells, and they differentiate into neurons. There's a lot of discussion we can have about this technology. It's all very new and changing every day, but it's a remarkable technology. And this piece was given a Nobel Prize last year, in fact. So if we add to the genomes the information we can get from the neuronal transcriptome, can we learn more? When by transcriptome, I mean the genes that are being expressed within these neurons. So first we look at these neurons here, these neurons that came from an individual, and we looked at what genes are present there. Well, they look a lot like neurons. They actually have all these neuronal marker genes in them. They look more than just like neurons. They also make astrocytes. They also make oligodendrocytes. And there in the culture, there remains some neural stem cells. The precursors of stem cells are also present still in that dish. So this is a complex culture. We're not going for a simple culture. We're going for something that recreates the milieu of the brain. Furthermore, this culture contains, looking at all the RNAs that are expressed, the mRNAs, this culture contains a wide variety of mRNAs from many different neuronal subtypes. Look at this. Here are all the glutamate ionotropic receptors here, NMDA, AMPA. Here are the metabotropic glutamate receptors, muscarinic and nicotinic acetylcholine receptors, DOPA, GABA, serotonin. All of these neuronal cell types are present within our culture system. So it's clearly a complex mixture that allows us to see a diversity of things that are present. So now I'm going to, so we have, actually before I actually go into that slide, we're beginning to do those cultures from 4 individuals from Colombia, and we'll rapidly get more so we can better interpret the genomes. But our work has moved a little faster with another condition called progressive supranuclear palsy. It's a condition that's a little bit like Parkinson's disease, but it has tau in the brain instead of synuclein. And in this individual, there's a mutation that creates increased risk for this disease called A152T in the tau gene. So once again, take a biopsy, make these neurons from his skin cells, and here is here's the patient, and there is actually what we can do here is look at the patient right here. I'm sorry, A152T. This one here is homozygous for A. This is what would be the wild type condition. This would be what most of us have. This is what the patient has one allele with a T. That's a genetic error. And then what we do is we make these cells homozygous. We give him 2 errors. You know, you get a copy from your mother and your father. We make both of these alleles now a mutant. And we do it through a technique that's called a zinc finger replacement technique, where we can make what are called isogenic lines that carry all of these different forms. We can also correct the mutation as well. And when you look at these pictures, this is stained with tau. Here are nice axons. The individual with this heterozygote mutation has disrupted axons, and in the homozygote, it's even worse. And I don't have the picture here, but we're just getting this data that when it's corrected with this zinc finger technique, when this is corrected, you can go back to nice-looking axons. So we can recreate at least some in vitro feature of the disease in a dish and correct it through a gene replacement technique. So here is the, what we're talking about here is the integration of cellular phenotypes and whole genomes to reveal causal variants. How can we put this information together. Most genetic variants with significance in all of these GWAS studies are in the non-coding portion of the genome. So how can we interpret that? They're not really right within protein regions. They're hard to interpret. But when we look at the expression level of the mRNAs, variations in the expression of mRNAs, which are called EQTL, stands for Expression Quantitative Trait Loci. These can pinpoint causal GWAS. That is, if you see an RNA that's increased, and upstream from that RNA in the non-coding region is a variant, then you might be able to zero in on that region. So here's our pipeline. We do the EQTLs and look in the iPS-derived neurons by sequencing them. They're deep sequencing. And we have full genomes on these individuals. So here are two individuals, one who has the A152T but lacks any phenotype, and this one has the disease. In fact, these two are brothers, and for some reason, he got it and he didn't. This is not like the disease in Colombia where everyone with the mutation gets it. This is not a fully penetrant mutation. So these are most people, if we compare any two people in this room, there's about 3 million differences between each of us in our genomes. In this case, they're brothers, so it's a slightly fewer number. There are 2 million differences between these two brothers. Then we do the RNA-seq, which means we sequence all of the RNA in their iPS-derived neurons, not just in the brother, but in a number of other cells we have growing in the lab, and we look for which genes are differentially expressed. There were 577 of them. And now we look around these 577 genes, and we found 150,000 genomic variants that are near these differentially expressed genes. We reduced the number from 2 million to 150,000. Now we do a thing that I sort of like to call gene gazing. This is just a sort of a pastime when you're like, after you finish a hard day's at work and you've had dinner and you just want to go and relax, you look at these genes and you just see, oh, that's a cool looking gene. So we look at these genes here and we find that some of them are actually these variants are in very interesting locations that we can further work up. Some of them are actually encoding variants. Some are them in highly conserved sites that are nucleotides that have been conserved back to rodents and earlier. Some of them are in microRNA responsive elements. Some of them are transcription factor binding sites, DNA methylation sites. Some of them have a very low minor allele frequency. That means they're rare changes. And, oh, thank you. Thank you, Jesus. And so this helps us to identify which of these non-coding variants may be more interesting and we should test further. The last couple slides is I want to show you one more mystery, one more paradox that we are facing in Colombia, because it's the Alzheimer family that we have studied there is actually only the tip of the iceberg. It turns out that there are a lot of other families that also have rare mutations. Here is a list of some of them. Familial Alzheimer's disease, this is the one I talked about. But there's another family in a different area outside of Medellin called Copacabana that has a different mutation in the same gene. Another large family, not quite as large. Here's a disease called CADASIL, in which people get strokes at a very young age and have mutations in Notch 3. It's quite rare. 2 families with this rare mutation there. And other rare conditions. For instance, this disease in which people get blepharospasm, and a rare pain syndrome with another mutation. These are autosomal dominant conditions. All of us are familiar with the fact that relatively inbred populations can get recessive conditions. But why would we see dominant conditions in this population, and so many of these rare dominant conditions? Antioquia has a population of about 8 million people. Most of those numbers of those people have come only recently because the population has exploded. And that's an important point. And I'll say more about that in a moment, but I just want to first show you what we can do with all of those mutations is to create a disease map. The red is where the Alzheimer families are, but there's other families down here in the southern part of the state. All of these colors indicate different autosomal dominant conditions and the areas where those families live. The people don't move very much. They stay in one place. So you can actually create a genetic disease map that, in which the geography coincides with the genetics. So now I want to make a final statement about why we may be, a speculative statement about why we may be seeing so many of these dominant conditions there. And I'm to introduce this last piece in the last slide or two, I want to quote a paper that is not something we did. I don't even know these authors. They were studying diabetes, and they took two genes implicated in diabetes, and they sequenced these two genes in over 13,000 people. And what they saw actually was that the number of variants in these genes kept, as they sequenced more people, it kept going up and up and up. It never really stabilized. If the population is stable, eventually you're going to get to a point where the number of mutations that we find as we sequence more and more people would level off. But that's not what's happening. That is that there's been this linear increase here in these in mutations as we sequence more and more people. And part of this comes from this explosion in the population. Here's what's happened in the population in Antioquia. It's compared to Costa Rica, which is actually very similar. Population has gone up quite a bit and very, very rapidly. Here is the census data from the towns that we studied here, in which you can see in Angostura, Medellin, Yarumal, this increase here in more recent years, 1928. Explosive growth in population can create what we might call a kind of saturation mutagenesis in the human genome. There are over 7 million people on the earth. There are 3 billion nucleotides in the human genome, and we are close to the point that every nucleotide that can be mutated has been mutated, except those that are lethal. And hence, the number of mutations that we can find keeps going up. So this recent explosive population growth, it may explain why we're seeing some of these concentration of dominant conditions in a very rapid growing population with preponderance of short recent genealogical branches. Most disease mutations have only arisen in the last 5 to 10,000 years. 42 to 75 de novo mutations occur in each of us. And 75% of de novo variants, here's another point, are from your father. These sperm cells are mitosing and mitosing and mitosing all through life. Egg cells, the ones women are born with, are the ones they have for life. They're not mutating as rapidly as sperm cells are. So older fathers are conveying more mutations to their offspring. If you have a family like those in Colombia with 15 children, the father by the 15th child is already going to be fairly old. So that may be another contributor to the genetic burden. So that is a final speculative reason. I want to now conclude here with this final slide about our work in Colombia. Every time I go down there and I see the people with dementia, I'm reminded of Macondo, Gabriel Garcia Marquez's story, 100 Years of Solitude, where he described this village of Macondo where all the people lost their memory. And if you remember that first chapter in his book, after the entire village had all this memory trouble, one day a gypsy showed up and brought with him una sustancia de color apacible, a substance of a gentle color, and cured everyone. Gracias, and thank you to all of the people who have helped with this enormously. I can go through all of these people for the sake of time. I'm only going to mention one person Francisco Lopera, who has been working tirelessly on this population for longer than I have. I've been there for about 25 years, and he's been there for 35 years doing this work. Here's the research team in Colombia. Here's a children's group that we've recently formed of the children from these families out here on Lopera's farm. And as I say, thank you to Dr. Lopera. Thank you all very much.
Thank you very much for your excellent talk. And we have time for 2 questions, short questions. Larry.
Why is the homozygote normal, given the prevailing hypothesis that these mutations are loss of function?
Yes, if you didn't hear the question, why?
Could you repeat it?
Yes. His question was a good question. He said, why is the homozygote normal when the hypothesis is that the mutations are loss of function? So there are a couple ideas about how the enzyme gamma secretase works. And one of them is that the mutation creates an alternative cut site in which instead of cutting at position 40, you cut at position 42 and creates something more toxic, and therefore you have a mixture of, say, of 42 and 40, and just more 42 if you have homozygosity. Maybe it's not lethal. The bottom line is I'm not going to be able to answer your question, but I would say that the speculative pathways here are, one, is the mutation working by shifting the cut site and generating more A beta 42, which may or may not be lethal? But the second idea, and why you probably said loss of function, is the other notion about how this enzyme works, which is that the enzyme works in a processive manner. That is, it trims back from 46 to 44 to 42 to 40. And that when we speak of loss of function now, and this is the way I think most people use the term, is that loss of the processivity, not really just a shift in the site. So if it's loss of processivity, I can see that you can have a homozygosity with not a complete impairment, if that's what we mean by loss of function. It's not like wiping out a lethal gene. I mean, a critical key gene.
Yes, I was wondering about the progression of these diseases and whether your genetic studies in kindreds that are more homogeneous genetically would tell us something about the onset of the disease, the times at which the disease starts and progresses. I would like your thoughts about this relationship between temporality and genetic homogeneity in the genome.
Yeah, well, because of the fact that there is this extraordinary synchronization of everybody in terms of their onset, that was why it was appealing for doing this clinical trial, obviously. The clinical trial is designed to give drug to individuals before they get any symptoms. To get into the trial, you have to be at least 28 or 29 years old. And if you get any symptoms at all, MCI, for instance, you're excluded from the trial. So having this clear-cut trajectory, not just with conversion to MCI, but even the biomarkers, like the changes in the amyloid PET and the CSF-A beta and tau, all move and neuropsychological testing, all move in a very predictable trajectory. So any deviation from that trajectory will be very informative in terms of these temporal factors. I'm not sure I really answered your question. I think I've just sort of, I'm pointing out why we're exploiting that feature for a clinical study. Why it is exactly that way, which is probably more along the lines of what you asked, may have to do with the fact that this is a very highly homogeneous population. Not only genetically relatively homogeneous, but they have similar levels of education, similar nutrition, similar occupations, similar geographic risks. It's very hard to do testing on any Alzheimer population with this degree of homogeneity.